Welcome students to the online lecture series of BIDWAN classes, and I am your bio faculty. So today we'll learn a interesting, basic yet interesting topic in biology, which is polymerase chain reaction. Although we have heard a lot about it, is while COVID time we heard ki, we need RT-PCR report, and we relied more on RT-PCR report. So the PCR there which came was very important. So it's an important tool in molecular biology, which basically means to amplify the amount of DNA. So PCR, as you can read, stands for polymerase chain reaction. So in this, the polymerase is basically the DNA polymerase, and chain reaction is the reaction occurs in a chain. So after many cycles of this reaction, you get an amplified amount of DNA. So this PCR was discovered by Carey Mullis in 1985. He basically was working in a corporation where, as a technician, where he used to synthesize oligonucleotide, and there he got this idea of how we can amplify this DNA in vitro. So if we define PCR as it is replication of DNA in vitro. So it basically works on the principle of DNA replication, which happens in the organism or in the cells in our body, but it is in vitro as in it occurs outside the body. Okay, so this is in vitro, and it results in selective amplification, which means that a required segment or the segment of interest can be amplified using PCR. Now, the principle on which PCR works, it basically is the principle that on which DNA replication works. So during DNA replication, what happens? The two strands of the DNA, so DNA is a double-stranded. Now the two strands get separated, then the primers come, they anneal to the template strand, and then the DNA polymerase carries out the extension. This is what happens in the DNA replication. Inside our body, we have lot of enzymes which carry out these reactions. For example, separation of DNA is done by helicase enzyme, then there is single-stranded binding protein, then there is DNA polymerase which helps in extension, then there is primer or primase which helps in synthesis of primer. Now we get rid of all these enzymes which is found in the cell or in the body, and if we want to carry out this amplification of DNA outside the body, we can do it using this PCR technique. So the principle on which it works is basically, so the first thing is you separate the strands of DNA, the two strands of DNA has to be separated, and then we need primer, because we know that the polymerase enzyme cannot extend the template DNA, or it cannot synthesize the new strand of DNA until unless it has primer. So we need primers for the synthesis of new strand, and then so now the DNA polymerase, if it gets the DNTPs, it can extend the template strand and can create new strands. So basically, so let's say this is your template strand. Okay. Now both of these are template strand. This is 3' to 5', this is 5' to 3'. Although here it is shown that it is a new strand, but let's say there were two strands, both of them were separated. Now the primer has come and attached here, and then the DNA polymerase, which is your main replicating enzyme, what it does, it replicates, it extends the primer, it extends the new strand with the help of primer, and then synthesizes the new strands. Now we'll see how polymerase or PCR works. So basically there are 3 steps in PCR. So first one is denaturation. What does denaturation means? Is that you need to denature the strands, both the strands of the DNA. So in PCR it is done, so it's basically the melting of the target DNA. So in PCR it is done at a higher temperature. So if you provide them higher temperature, So if there are two strands, if you provide them higher temperature, then they will get denatured. Now we have to keep in mind that the temperature should be provided only for certain amount of time, so that these hydrogen bonds which are present between the nitrogenous bases needs to be broken down. So now as we provide the higher temperature, which is usually 94 to 96 degrees Celsius, the two strands will get separated. Okay. Now each single strand will act as a template strand. So both these strands, both the single strands will now act as template strand. Okay. Now the next step is the joining of primer or the annealing. Anneal means to join. So as I told you that the DNA polymerase enzyme needs primer for the extension of new strands or the extension of the template strand. It cannot initiate the replication, or it cannot add the DNTPs on its own. So we need to synthesize, or we need to attach primer to the template strand. So what we do is we have two primers, because there are two single strands. Those primers get attached to the 3' end of the template strand. Okay, so let's say this is 3' to 5' strand, and this is 5' to 3' strand. So at 3' end, the primer get attached. Okay. Now this happens. So how we join the primers to the single strand is हम temperature को low कर दे देते हैं. So in denaturation the temperature was high. So at high temperature they get separated. Now as we reduce the temperature, जब हम temperature कम करते हैं, usually it is at 40 to 60 degrees Celsius, what happens, the primers get annealed to the single strand. Now this primer will have free 3' OH group. Okay, so this is our primer. So it will have 3' OH group where the DNA polymerase can extend the new strands. So these primers are nothing, basically short single strands of DNA, which is called as oligonucleotide. Okay, so we need two oligonucleotide. One is to attach in the 3' end, the strand which extends in 3' to 5' end, and the other one in which the strand extends from 5' to 3' end. So after this annealing of primer, now the last step which is there is to elongate or polymerize the chain, or polymerize the strand of the DNA. So this is done by enzyme which is DNA polymerase. As we know that even in our body, DNA polymerase ही length को extend करता है और the strands को extend करता है. So now this reaction is carried out at a temperature which is 72 degrees Celsius. So what happens at this temperature, the DNA polymerase, and we provide the DNTPs, what DNTP, the buffer to carry out the reaction to extend the length of the DNA. Now the DNA polymerase which is provided here is basically the tagged DNA polymerase. What we know is that कि हमारे body में जो temperature है is basically the temperature which is usually less than the 72 degrees Celsius. Now that DNA polymerase cannot act at this high temperature, it will degrade. So we have isolated a DNA polymerase from a bacterium known as Thermus aquaticus. Okay, so this bacterium is basically a thermophilic bacteria. It stays at a very high temperature. For example, 100 degrees Celsius hot springs pe it stays. So we have isolated this Taq polymerase enzyme from this bacteria. Now this can act at a higher temperature. So using this Taq polymerase enzyme, using DNTPs, which will be required to extend the length, the strand of the DNA, and the buffer, the length, the template strand is extended. So this is a kind of summary where you can understand how the whole polymerase reaction goes on. So this is your two template strands. Okay, what we do, so first step was denaturation, जहाँ पे we provide higher temperature. Now this higher temperature, what it will do, it will denature the template strands. So now these two single strands of DNA are created or they are separated. Now next step is the annealing. So annealing mein what we do, we lower down the temperature, which is usually 40 to 60 degrees Celsius. यहाँ पे 68 लिखा है, but it can range, it can vary from 40 to 60 degrees Celsius. So what we do in annealing is basically the primers gets joined to the template strand. Okay, both the template strand, the primers get joined, and they are named as forward primer and let's say reverse primer. So the two oligonucleotide primers get annealed to the single strands of the template DNA. Now the third step is the elongation, which is carried out by Taq polymerase enzyme. This enzyme works at higher temperature because it is extracted from a bacterium which can survive at higher temperature. So now what it does, so as we know that if a primer is there and DNTPs are there, so now your DNA polymerase can extend the template strand or synthesize the new strand. Now this whole cycle is carried out several times. So it is basically a chain reaction. So it is carried out for several reactions, and at the end of many reactions you will get large copies of DNA, and you get the amplified DNA, which is your DNA of interest, which you want to amplify. Now there are several, so now this is PCR, okay, this is what PCR and how it works. So basically if you have very small amount of DNA and you need to amplify it, आप उसको बहुत times amplify करना चाहते हैं, and you want to do that, so there is a very convenient technique which is called polymerase chain reaction, which can do this. Now the question comes कि why is it required? हमें क्यों ज़रूरत है that we need a large amount of DNA? So there are many experiments, there are many things which we can do by amplifying the amount of DNA, and for that we need a large amount of DNA. Okay, so for example, I was in initial when I introduced, मैं बता रही थी कि during COVID time it was asked, आप कहीं भी जाइए, it was asked, we need your RT-PCR report, we do not rely much on your antigen or antigen report. तो क्यों? Because क्यों they were relying more on the RT-PCR report or the PCR report? Because there even a small amount of अगर virus भी है किसी के अंदर, in our body, अगर बहुत कम amount भी है virus का, so the PCR, if we get that that DNA or the virus का DNA, if we can get it, हम उसको amplify कर सकते हैं in a large amount, and then we can detect कि yeah, virus is there, and therefore you have the disease, you can have the disease. Even अगर बहुत small amount भी है, that also can be amplified. So there are many applications of PCR. Some of it we can look at it, for example, if we need to detect any pathogen, just like I told you, here HIV virus or the COVID virus, okay, if you want to detect any pathogen and it is there in very small amount, even at the early stage, let's say the disease at a very early stage, you do not get the symptoms of it, but you want to detect it, so you can do it using PCR, because even the a very small amount of that DNA can be amplified to a greater extent, and we can detect the pathogen. For example, other is DNA fingerprinting, is a very important technique again, where we need the DNA of the test organism or the test individual and the other individual, and if we are making a DNA profile, वहाँ पे भी we need DNA, so we need a large amount of DNA. So there also the PCR, using PCR you can amplify that amount of DNA. The other application is prenatal diagnosis. So for example, कि a child is about to born or is in the womb of the mother, और उसको कोई disease, ऐसा genetic disease है which is very deadly, and the doctor wants to confirm whether it has the disease or not, whether child will be born with a disease or not. So what they can do is, even before taking birth, the genetic disease can be detected by taking that DNA and then amplifying it in a large amount. In paleontology also, for example, we have fossils of, fossil remains of plants and animals. So there, we can study those fossils, taking their DNA, amplifying their DNA, and then studying them. Also in gene therapy, monitoring the gene in the gene therapy also. When we'll discuss what is gene therapy, I can tell you in detail what is gene therapy and where. But wherever you need a large amount of DNA, we can use PCR and get a large amount of DNA. I hope ki what I told you makes a good sense, and you can understand what PCR is, what are the basic steps in PCR, and on what principle does it work. Okay, so very important is to understand कि PCR किस principle पे work करता है. Okay, and then if we understand the principle, then we can get the whole technique, how does it work and what does it stand for. I think I have made you understand enough what is PCR, and if you like this video, please like it. If you want other, your friends to know what PCR is, you can share it, and also don't forget to subscribe our channel. Thank you.